Thank you so much, Bethany. Really appreciate it and love this opportunity to talk about climate adaptation science. I'm guessing that most folks here will have a sense of where the Boston Harbor Islands are. There's 29 islands and 5 peninsulas in the park, in the urban Boston area and in or just outside of the Boston Harbor estuary. As just noted in the introduction, the Boston Harbor Islands are part of a three national park unit we call the National Parks of Boston, including the other 2 parks that Bethany just mentioned. Just a little bit about some of the sort of things you might see if you're flying above or visiting the Boston Harbor Islands. A lot of what you can see on the landscape are 18th, 19th, and 20th century features related to navigation and coastal defense. There's the Boston Light National Historic Landmark out on Little Brewster Island, the Fort Warren National Historic Landmark on George's Island, and the islands have been subject to quite a bit of disturbance over the past few centuries. The images there of Gallops and Peddocks Island, which are 2 of the islands that are part of this study, underscore how much alteration there's been to some of the landscapes over time. Those are from the early 20th century time of the Army being using those islands. The islands also have quite a long post-colonial history of uses for various institutions, the kinds of things that were either not wanted in Boston or made sense at the edge, such as quarantine stations and hospitals and associated cemeteries, sewage treatment facilities, and things like brothels and gambling houses and horse rendering plants and all those kinds of things that the proper Boston, as it was developing as a major city, didn't sort of want right in the city. The park now is an important refuge for coastal biodiversity, particularly since it is comprised mostly of islands for coastal breeding bird species that are relatively free of mammal predators. Also intertidal habitats, including rocky intertidal and salt marshes, and we'll be talking a lot more about that. Folks can visit the Boston Harbor Islands between Memorial Day and Indigenous Peoples Day. There's public ferries that go out to 4 of the islands. And this aerial image is more or less how Congress directed park managers to think about the park as an integrated landscape, including the islands and the surrounding mainland area and the marine ecosystems. So our responsibility really is to think of it as a whole, because of course, birds and fish and intertidal critters don't see the distinctions between the urban areas or what town things are in or anything like that. One of the nationally significant features of the park that is part of the justification for being part of the national park system is that the Boston Harbor Islands represent the only partially drowned drumlin field in North America. So drumlins are essentially glacially sculpted hills that through the advance and then retreat of the huge ice sheets end up getting sculpted into sort of elongated oval shapes that are indicated there in the graphic. And many of the harbor islands are comprised of drumlins, and it's part of a drumlin field that intersects the coast. So there are many of the drumlins that are part of the same field are currently on the mainland. Now this is important because the glaciers move sediments in a way that's very unsorted. You know, they move boulders just as easily as they can move little clay particles. And that left us with an interesting landscape in the harbor islands. And I wanted to talk a little bit about the sediment particle size and the habitats that emerge from that. On the bottom, you see the arrow bar distinguishing, with a gradient from smaller to larger particle size sediments, from the tiny clays and silts up to the very large cobble and boulders and even permanent bedrock. And what you end up with habitat-wise are salt marshes in the areas that are more quiescent, where the small, finer particles can settle out of the aquatic system and basically feed the plants. And on the extreme other end, you end up with more classic rocky intertidal habitats, the sort of permanent intertidal that you think of when you think about tide pooling. And in the middle are all these sort of glacially moved sediments that formed what are called mixed coarse intertidal habitats. And that's the focus of this work. This was inspired by an important inventory done in 2003 by Rich Bell and collaborators, which identified this mixed coarse substrate as essentially what it sounds like: mixed up materials that include some larger materials. And here's a map showing some of the data from that Bell survey, just for Pettix Island, which is one of the focal islands for this study. And the yellow portion of the bars is the mixed coarse substrate on the different islands, and you can see that how significant a proportion of the habitats across these islands mixed coarse substrates make up. Now, as park managers, of course, just like everyone who's trying to manage parks in this time, we are dealing with changing ecosystems, changing landscapes. In our case, coastal landscapes. On the left is Lovell's Island, with some of the Army infrastructure sort of having fallen out of the bluff. And on the right is a wall of cobble that got moved by the March 2018 nor'easter storm out on Calf Island. Of course, on top of and within the islands themselves, there's sort of innumerable stories related to the cultural heritage of this area. On the top left, there's George's Island again with Fort Warren, and you can see the erosion directly affecting the earthworks there. And there are dozens of known and more unknown sites associated with indigenous cultural heritage on the islands. And each storm that erodes the bluff has the potential to erode some of those creations without us even knowing it. As inspired by that, the city of Boston has begun a project that we are part of called the Boston Harbor Islands Archaeological Climate Action Plan, which is intended to prioritize sites that might be most likely to erode soonest, and then include indigenous partners in the decision making about what to do about that. So most folks here probably are familiar with sort of park science and parks and how it works, but basically science is intended to help us make smart management decisions to take care of parks. The reason why we have national parks is to preserve the important natural and historic features of these places for all future generations to enjoy. And we need good information to do that. The managers in this case is a consortium of various partners. The map on the top right shows the 9 different landowners that own part of the Boston Harbor Islands National and State Park. The Park Service owns just a very tiny proportion of the park. We do, however, have a role in standing up the Boston Harbor Islands Partnership and helping to facilitate collaborative planning and management, such as scientific studies like this. Now, that graphic on the left is representing the federal partners, the City of Boston partners, the state partners, and nonprofit partners, as well as the Public Advisory Council. And like other parks in this day and age, we are trying to be climate smart with planning, sort of future planning for how to conserve our park landscapes and ecosystems. And we hope that studies like this will help us consider alternatives within this resist, accept, direct framework, where we can be thoughtful about how we want to address the predicted effects of climate change, whether we want to, say, build walls and resist higher water in our case, or whether we might want to accept higher water flooding certain areas, or whether we want to maybe direct some of that higher water in a way that is strategically wise in terms of conserving the most important things in our parks. Part of the toolkit that we are hoping to develop would be nature-based solutions, which are projects that work with nature to address sort of impacts, in our case, of coastal erosion and coastal flooding in a way that provide co-benefits, ecological co-benefits or public co-benefits for recreation. Fortunately, we have right in our park a research and education partnership called the Stone Living Lab Partnership that is intended to do just that. And we work alongside the city and the state, UMass Boston, and Boston Harbor Now to try to facilitate that. Okay, so this project in a and but therefore framework. So here's our challenge. We must try to conserve the parklands that have historic and cultural and natural values, given that we're now experiencing climate change, while also considering the effects of any projects we do on biodiversity. The islands have this unusually high percentage of mixed coarse substrates, but the methods for biodiversity surveys in permanent rocky intertidal habitats may not be best for effectively tracking biodiversity in these mixed coarse sites. Therefore, this project was developed to determine if we can efficiently and reliably inventory the biodiversity in these sites on those habitats. So the major activities of this project involve a synthesis and an inventory of all the existing information so we know where we're starting from, developing a standardized, repeatable monitoring framework, and then using that framework to collect baseline data that will then allow us to measure change in the future. The focal areas for this project are some of the larger sort of middle harbor islands that have docks and piers, important historic and natural features, and recreational amenities, because these are the islands where we're most likely to have adaptation projects happening, and also islands with a lot of mixed coarse substrate. And with that, I'm going to turn it over to Aly to talk us through what we've been doing.
Thanks, Marc. Now that we've outlined our methods and timeline, let's take a look at the key outcomes of this project and what we've learned so far. Next. This slide provides a roadmap of our methods starting in 2020 during COVID, where we conducted a historical inventory of species records and then gathered info on these species' vulnerability to climate change. In 2021, we walked the shorelines of many islands to characterize habitats and identify sites to test the effectiveness of new methods in 2022. And then in 2023, we collected baseline data across multiple islands and sites. Next. To better understand the current state of biodiversity, we first looked at historical data to see how species diversity has changed over time. Next. Before beginning our fieldwork, we developed an inventory of marine species in Boston Harbor, focusing on species taxonomy, including introduced species, and conducting a biodiversity analysis to better understand the ecosystem. This paper was most recently published, open access, in the International Scientific Journal Marine Biodiversity, and we suggest you give it a read. These plots here show the number of unique species detected in Boston Harbor since the late 1800s, based on records from reports, scientific publications, and community science platforms like eBird and iNaturalist. Over 450 species have been documented across the Boston Harbor Islands, with intertidal invertebrates and algae being the most diverse. We see three key phases in increased species detection over time. First, harbor-wide cleanup efforts and establishment of state park in the 70s and 80s. Second, the establishment of National Park Service and their management in the 90s, which boosted scientific research through directed and facilitated efforts thereafter. And finally, digital platforms like iNaturalist and eBird, which have made recording species easier and more accessible. However, in recent decades, the trend towards higher biodiversity has also been driven in part by detections of non-native and invasive species. Some species are already here and have been here for a long time, including some species of crab. Next. Like green crab and Asian shore crab, seen here in this video that we took on Lovell's Island. These are Asian shore crab, highly invasive, and are one of the dominant crab species in the intertidal across the islands and in New England. Okay. Oops. In our biodiversity analysis, we used a metric called average taxonomic distinctness, also AVTD, which you'll hear me say over the next couple slides. This measure helps us assess biodiversity across different time periods, even when sampling efforts have varied widely or are incomplete or incompatible, like comparing today's data from iNaturalist with limited records from the early 1900s. AVTD provides a standardized way to account for these differences using the taxonomic distance between species to capture overall biodiversity. Higher AVTD values indicate greater biodiversity, and lower values suggest a lower biodiversity. This graph shows the average taxonomic distinctness by decade for all species found in Boston Harbor from 1861 to 2020. We found a significant difference in ABTD scores across the decades. On average, the later decades, the 1990s through 2020, had higher ABTD values, including indicating greater biodiversity compared to earlier decades. These results align with the conservation efforts that began in the 1980s to improve water quality. For example, since the early 1980s, dissolved oxygen levels in the harbor have steadily increased, largely due to cleanup of wastewater discharge, which had previously caused eutrophic conditions. This graph shows the relationship between average taxonomic distinctness and dissolved oxygen levels in Boston Harbor. We found a significant positive correlation, higher ABTD values indicating greater biodiversity were associated with higher dissolved oxygen concentration. Dissolved oxygen is crucial for the survival and growth of many marine animals, including fish, crustaceans, and bivalves, and plays a key role in supporting healthy ecosystems and promoting biodiversity. For example, eelgrass, Zostera marina, which is crucial for juvenile fish habitat and carbon sequestration, has shown increased abundance in Boston Harbor. This resurgence is linked to the rise in dissolved oxygen levels, a direct result of the cleanup efforts in the harbor. We found that low values of ABTD, indicating low biodiversity, were associated with high sea surface temperatures. This relationship is consistent with well-documented climate change impacts, where increased sea surface temperature leads to a decline in the abundance and diversity of native species that are vulnerable to climate stress. As Boston Harbor faces increasing threats from climate change, including rising sea levels and coastal erosion, it's crucial to assess the biodiversity in preparation for adaptation efforts. But before we can properly assess, we first need to develop the right protocol and tools to effectively monitor and track these changes. So we created a new protocol for mixed coarse substrates by testing various methods at 3 types of sites: erosion-prone areas, high biodiversity habitats, and control sites. By comparing species detected and the effort needed, we refined the approach and used it to gather baseline data across 5 islands and 10 sites. Before I get into our methods, I want to first reorient you to the intertidal zone. This is the space that experiences both inundation by water at high tide and exposure to air at low tide. A typical rocky intertidal has clear zonation patterns that host unique communities of species with different tolerances to exposure of air and seawater. These zones span a gradient of high, mid, and low, and biological zones that are characterized by distinct bands of red and brown seaweeds and habitat-forming invertebrates such as barnacles and mussels, as you can see in this photo. And these general patterns occur globally on rocky intertidal shorelines. In comparison, here I'm showing you a mixed coarse intertidal, one of our sites on George's Island, where we still have zonation patterns. However, they're a little less uniform and more dynamic in space due to the mixed coarse substrate. So given this dynamic and less uniform zonation in mixed coarse habitats, we needed to carefully consider which monitoring methods would best capture the full complexity of these sites. Existing protocols may focus on indicator species, which are those that broadly represent ecosystem health. That approach allows us to track changes without surveying every species, but it may miss shifts in overall biodiversity, especially in complex multidimensional habitats like mixed coarse substrate. In contrast, community-level monitoring captures entire species assemblages, giving a fuller picture, but requiring really detailed baseline data. For variable sites like ours, this baseline is essential to track changes accurately. A key challenge is adapting these methods to mixed coarse substrates, where loose materials and variable stability can impact reliability. We weren't sure if existing methods would be effective, so testing multiple protocols helped us to find the best approaches for capturing the complexity of mixed coarse substrate. High spatial coverage is crucial in mixed coarse substrates where species distribution are patchy and variable. Our protocol needs to balance broad habitat coverage with targeted detail to capture cryptic species that play ecological roles but may be missed in surveys. For example, here we see an invasive orange-striped anemone under a boulder on Gallops Island on the left, and a dwarf brittle star hidden between cobble on Lovell's Island on the right. Finally, protocols need to adapt to habitat complexity, especially in these diverse environments of mixed coarse substrate. These habitats vary greatly across even short distances, so protocols must be adaptable and flexible enough to capture species across different zones and substrate layers. Here I'm showing you two sites on Gallops Island. Both have mixed coarse substrate, but as you can see from the zonation colors, the biological zones differ. The sites also vary in vertical shoreline length. From top line to low water at low tide, the site on the left spans 18 meters, while the one on the right reaches out to 55 meters. So we tested 3 established protocols commonly used in the Northeast, each focusing on different components of rocky intertidal systems. The Northeast Coastal Stations Alliance, or NECSA, the National Park Service Temperate Network Long-Term Rocky Intertidal Monitoring, or NETN, and the Stone Living Lab, provided to us by Dr. Jarrett Burns at UMass Boston. Each protocol divides the shoreline into zones, high, mid, and low, and uses a series of transects and quadrats to survey species across these areas. These protocols differ in grid layout, quadrat placement such as random versus fixed, and inclusion of biological zones, and species identification intervals along transects, also known as point intercept. Each method we tested offers a distinct strength. The Stone Living Lab for broad spatial coverage, NETN for focusing on well-defined biological zones, and NECSA for structured, delineated sampling and point intercept. However, do these protocols fully address the unique challenges of mixed coarse substrates where species may inhibit very specific layers or areas? We wanted to learn more. Given these strengths and variations, we thought a hybrid approach may be promising. By combining protocols or components of the protocols, we could leverage each method's strengths to create a more comprehensive picture of biodiversity. So we tested all 3 protocols to understand how well each would be adapted to mixed coarse substrate. We gathered the information on how the protocols handled species in varied substrate types, from stable rock to loose coarse material, and where biological zones may be patchy or inconsistent to inform our next steps. In the summer of 22, we conducted surveys on Georges Island, focusing on 3 distinct sites to evaluate and compare the methods of these 3 protocols. Testing all 3 protocols at each site allowed us to assess their effectiveness on this island's unique mixed coarse substrate and determine which elements worked best for our new monitoring approach. The 3 types of sites selected for testing the protocol were sites of cultural importance and management relevance to the National Park Service, including those considered for restoration or adaptive coastline management due to critical infrastructure such as seawalls or historical landmarks at high risk of erosion, as seen in these photos. Biodiversity hotspots, which include sites near other coastal habitats such as salt marsh, rocky outcrops that edge into mixed coarse areas, and had keystone, indicator, or foundational species such as mussel beds, eelgrass, and fucoid macroalgae. And finally, control sites where generic mixed coarse substrates without significant anthropogenic manipulation or nearby structures, and without keystone or foundational species present, they in general looked bare. Here's a photo of Georges Island, high biodiversity site, where you can see dense fucoid canopies along the broken seawall, patches of mixed coarse and cobble, and what's covered by the water, this photo was not taken at low tide, were other important species such as dense mussel beds and more fucoid stands. So here I am showing you the number of species found historically on Georges Island with the addition of our method testing in 22. Our protocol testing revealed a significant increase in species diversity compared to previous records on the island, highlighting the enhanced effectiveness of our updated sampling methods. So from our protocol testing, we identified some key insights to help us develop and implement our new set of methods. First, site selection. Our results showed that predetermined high biodiversity sites captured more species diversity than generic or erosional sites, which were found to be similar. This allowed us to simplify the framework by keeping our NPS designated sites while eliminating the generic site, reducing our effort while maintaining data quality. We compared 3 protocols to see how many species they could capture. By using species accumulation models, we found that the NETN and the Stone Living Lab protocols identified the most species, highlighting their effectiveness. The Nexa protocol had lower species richness, so we focused on adapting the NETN and Stone Living Lab for better monitoring. We also found that quadrats, which provide more detail, were far more effective than point intercept methods. Because point intercept took more time and effort without adding much value, we decided to remove that from the final protocol and focus on quadrats for better coverage. Finally, our analysis showed that both biological zones like ascophyllum, barnacle, fucus, mussels, and red algae, and protocol type NETN and Stone Living Lab were strongly linked to species patterns. Key factors like randomized quadrat placement and transect line length also played a significant role. And so this helped us finalize a protocol that captures important ecological patterns across different habitats efficiently. We also piloted several novel tools, including crab traps and surveys, photo quads and image analysis, and environmental DNA to see if there were low-cost, low-effort options to collect biodiversity data that the park could use to create opportunities for citizen science, outreach, and education in the park. These methods are public friendly in that the traps and transects gather data on crab abundance and distribution, as well as catchability by either walking transect lines and looking under rocks and seaweeds, or placing traps and checking them after tidal cycles. Photo quadrats allow for images to be collected from quadrats that could then be evaluated remotely on a computer, and environmental DNA, which is a molecular technique that is rising in popularity, as a rapid method of looking for shed biological material from organisms in water samples. So on to our protocol testing. Next. In 2023, we used our newly tested protocol to gather biodiversity data at our study sites. For each site, we mapped a 48-meter section of shoreline and divided it into a 9-quadrant grid. Using a randomized sampling approach, we placed 36 square sampling quadrats across the grid, specifically taking into account biological zones. This setup allowed us to capture a clear picture of species presence and abundance in each zone. We surveyed 5 islands and 10 sites, including high biodiversity and NPS sites of importance. Using our new protocol, we visually surveyed with quadrats, conducted crab surveys with both band transect and traps, collected photo quadrat data, and gathered environmental DNA samples to assess biodiversity. Additionally, we took spatial data using real-time kinematic GPS to map the layout of each site and ensure that we can revisit quadrats in the future. We also deployed hobo loggers to monitor water and air temperature, salinity, and water height, critical data to track changes as sea level rises and to assess long-term impacts of climate change. These maps here show 2 sites on George's Island, highlighting the differences in where certain habitat zones are located. Some zones are present in one area, but not the other. This pattern is true across all the islands we studied. The habitat zones and the species that live there can tell us a lot about how the environment is changing. And by tracking these biological zones, we can better understand how climate change is impacting the islands and guide efforts to restore and protect these habitats. Our RTK data was essential in creating these maps, allowing us to accurately capture the spatial layout of the sites and track changes over time. Using Lovell's Island as an example, this plot compares the number of species recorded historically from the earliest observation in 2001 to 2019 with the number detected using our protocol. As you can see, our methods identified significantly more intertidal species than previous efforts. We have similar data like this for all 5 islands where our efforts were much higher than previous recorded, but for the sake of time, we're just showing data from this one island. We also found that some of our additional methods, in this case our crab surveys, varied in their adaptability to detect different native and non-native crab species using transects and traps, with the transect method being better to detect invasive Asian shore crabs and native hermit crabs, and the traps being better for detecting invasive green crabs and native cancer crabs. These methods are very public friendly and could be implemented to engage and educate park visitors and student groups on invasive species in the Boston Harbor Islands. This graph shows the number of species found on each island that we sampled. Gallops had the highest number of intertidal species, followed by Georges, and Pettix, Lovels, and Thompson had similar totals. Given species present at Gallops is especially interesting. The limited access to Gallops Island over our 150-year record only has 5 species documented, but our efforts increased that number from 5 to 60, which was really exciting. And although not found directly in our quadrats, Gallops Island actually had important species not seen on other islands, such as sea stars, which are believed to be in decline right now. We didn't find just one, actually, we found several. So those occurrence points give us hope on the sea star front. Here I am showing you the same graph I previously showed, total number of species per island, but I'm breaking it down by the number of species and phylum they belong to. A few interesting points. Although Lovels Island had a lower number of unique species count, it actually has higher representation across the phyla. There are species from 10 phyla. The others islands only have about 6 to 8 represented. Lovels also is the only island to have found a species from the Echinodermata family. This is Amphiphilus squamata, also known as the dwarf brittle star. It has only been recorded on one other time in Boston Harbor, and that was back in 2007. Similarly, Gallops is the only island to have found a species from the annelid family. This is Lepidonatus squamatus, the 12-scale worm, found only twice here in the harbor at Rose Wharf through the Massachusetts Coastal Zone Management's Rapid Assessment Survey in 2013 and 2018. Another key method we used is environmental DNA, or eDNA. This technique involves detecting DNA that organisms naturally shed into the environment, in this case, from intertidal areas. Cells from skin, waste, and other materials get left in the water, and by collecting water samples, we can analyze this DNA to identify species present. At each of our sites, we collected water samples, extracted and amplified the DNA, and identified the species found. Our goal was to test how effective eDNA for detecting intertidal biodiversity was. Here we compare species detected by our visual quadrat surveys, the overlap with species detected by eDNA, and the total species identified solely through eDNA. For intertidal species, visual surveys were more effective as eDNA only captured a small portion of these species. However, eDNA identified a much wider range of species overall, particularly microbes, plankton, and fish, including sturgeon, and ecologically important species that may use the intertidal at high tide. This shows that eDNA, while not a standalone replacement for visual surveys, is a useful tool to complement traditional methods by providing a broader picture of the habitat and species that interact with the intertidal zone. Our project set a strong foundation for future biodiversity monitoring of mixed coarse habitats in Boston Harbor using a combination of innovative and tested methods to capture a broad picture of intertidal life. Our new protocol worked well in capturing biodiversity baseline across the islands, providing data for future work to build on and conservation efforts. Predetermined by high biodiversity sites confirmed our expectations, showing greater species richness. Different methods of crab detection varied results on crab numbers, highlighting the importance of using multiple survey techniques for accurate data. And eDNA helps detect species that may pass through intertidal zones like fish and adds valuable information. However, it shouldn't replace visual surveys, but complement them by capturing additional species. And finally, photo quadrats allow us to document habitat structure and species composition over time and have a digital record to be able to track changes. So our project successfully established broad biodiversity data for Boston Harbor's intertidal zones with mixed coarse substrates using diverse methods from visual surveys and photoquads to eDNA. Really, the takeaway here is that these tools really complement each other, allowing us to detect biodiversity, track crab species effectively and provide a fuller picture of marine life, which is crucial for the future of conservation and monitoring efforts in this area. Now I'm going to pass it back to Marc.
Thanks so much, Aly. I'm just going to wrap us up here with a couple slides about some outcomes and implications from the sort of management perspective. And this is really just sort of restating a lot of what Aly was just saying there at the end. As I noted, our goal was to figure out whether we could develop good understanding about the mixed coarse substrate habitats and biodiversity in our park through a repeatable method. That's what we did. The baseline, we now have much greater baseline knowledge of the intertidal biodiversity. We understand that there's quite a bit of variation within islands and across islands. As Aly noted, even the banding of different intertidal zones is different within and across islands, and therefore we really can't use one index site or a few index sites to tell us the impacts of a given project on another site. And we do now have a protocol that we think can efficiently and reliably inventory the intertidal biodiversity of these mixed coarse substrates. So good science and the products associated with that. We have the visual survey protocol, we have the crab survey protocol, the photoquadrats protocol, and the eDNA protocol. We also have a very handy algae, or not algae, but intertidal species ID guide is the algae guide page of it, or the one of the pages of it, that will be useful for our park staff, whether they just are getting familiar with the intertidal habitats of the park, could be useful in education programs, and certainly would be useful for any future participants in similar surveys. And one of the questions we have is what to do about the fact that our vital signs monitoring, which is a Park Service sort of permanent monitoring protocol or program, includes the outer island rocky sites, so those permanent rocky intertidal sites, which we are getting very valuable information about. However, as described in this talk, we are also very interested in the information about these mixed coarse substrate sites, and we're not sure that the information from one habitat type is directly transferable to the other, probably not. So we have a question about the possibly adapting our intertidal monitoring into the future to be determined. From my perspective, given our urban partnership park context, probably equally important as the scientific information is building a network of knowledgeable folks who can help us who we can go to help understand what's going on in the park. In the case of the many students and interns that participated in this project through the three years of the field work, we have provided a professional development opportunity for them, and we expect to see one or more of them coming back around and being excited about working with us to learn more about these intertidal habitats. Also, working with these students, some of whom came in with very little knowledge, provided us a good baseline for understanding which parts of the methods are appropriate for a participatory or citizen science protocol that we might develop in the future. And the core team is represented here. Dr. Michelle Staudinger led this project team initially when she was at the USGS, and she's now more recently moved on to the University of Maine. She held us together and helped adapt our protocol, or adapt our sort of team approach during the COVID sort of shutdown of a lot of the anticipated initial work. And then you can see the rest of the team, in addition to Aly and me, from UMass Boston and USGS. And with that, we would love to hear any questions or ideas from anyone.